Have you ever wondered how scientists determine if a population is truly evolving, or how they measure genetic change over time? It is a fascinating question that requires a fundamental point of reference. The Hardy-Weinberg equilibrium serves as a fundamental genetic baseline because it describes a hypothetical population where no evolutionary forces are acting. Think of it as the perfect and changing scenario against which all real-world genetic shifts can be measured. It provides a crucial null hypothesis for population genetics, meaning it outlines what conditions would exist if evolution were not occurring. The core reason this principle acts as such a robust baseline lies in its strict set of 5 conditions, each designed to prevent any form of evolutionary change. First, there must be no mutation, meaning no new alleles are created and existing ones do not change. Second, there can be no gene flow, which means no individuals or their genes move into or out of the population. Third, mating must be random, ensuring that individuals do not select mates based on specific traits. Fourth, the population must be extremely large to prevent random fluctuations in allele frequencies, known as genetic drift. Finally, there can be no natural selection, meaning all genotypes must have equal rates of survival and reproduction. If any of these 5 conditions are violated, evolution is happening, making the Hardy-Weinberg principle the perfect standard for comparison. So, how do scientists actually use this baseline? They will calculate the expected allele and genotype frequencies for a population assuming it is in Hardy-Weinberg equilibrium. Then, they will compare these theoretical frequencies with the actual frequencies observed in a real population. If there is a significant difference between the observed and expected values, it indicates that one or more of the evolutionary forces, mutation, gene flow, non-random mating, genetic drift, or natural selection, are at play. This allows researchers to quantify and understand the specific evolutionary pressures acting on a population. This concept is incredibly powerful because it is not just about identifying if evolution is happening, but also about helping to pinpoint which evolutionary mechanisms are most influential in a given context. For instance, if a population shows a consistent deficit of a certain homozygous genotype compared to Hardy-Weinberg predictions, it might suggest heterozygote advantage or perhaps strong selection against that specific homozygous state. This deep dive into population dynamics allows for very precise insights. Ultimately, the Hardy-Weinberg principle provides an invaluable theoretical framework. By understanding this idealized state of genetic stasis, we gain the essential tool to identify, measure, and interpret the dynamic forces of evolution that constantly shape the genetic makeup of real populations. It is the gold standard for understanding how and why genetic change occurs.